Good morning. Good morning, everyone. Good morning. Happy Wednesday. Gloomy Wednesday. But we need the rain, so we need the rain to wash the pollen away. Good to see everybody. Yes, if you would, please go ahead and type your first name into chat for me. Thank you. Looks like just about everybody is here. We'll give folks just another minute or two to come on in. Lisa C, I think your microphone might be on. You might want to double check that.
Okay, I'm actually away on my trip.
Oh, you are?
Yeah, I don't have a very good connection in my hotel room. So I'm not sure if I can watch the video when I get home.
You can, absolutely. You can wait and watch the video on our playlist when you get home, or you can try from if you have the time.
No, I have the time. I set it aside, but I'm just letting you know it keeps going in and out on me. Yeah. So I don't know if I'm going to be able to sit through it.
Sure, I understand that. And remember that, at least in my experience, if you turn off things on your end, if you turn off your microphone and your video, it might stream a little more smoothly for you.
Okay, thank you.
Sometimes it's all magic as far as I'm concerned. So
it is. And my whole screen just disappeared. But yes, I will try that. Thank you.
Sure, no problem. All right. Okay, so today, what we're going to be doing is talking about another type of staining protocol, which I shared with you was the single most common staining protocol in use in microbiology labs, and that's called gram staining. The last time we were together, we talked about simple staining and the use of one stain to stain a bacterial smear and examine cells. The purpose of gram staining is a little bit different than simple staining. It's not just about being able to see the cells and tell what relative size they are and what shape they are. With gram staining, we're actually starting to classify cells. We use gram staining as one of the many tests that we run to help us identify a bacterium. So again, when we put methylene blue on cells and we can tell that they are cocci or rods or spirochetes, when we put methylene blue or another simple stain on cells, it doesn't tell us anything other than relative size and shape. We certainly can't identify what genus and species we're looking at. You can't. All you're doing is colorizing the cells. With gram staining, we're starting the process of identification. What gram staining allows us to do is place bacteria into one of two very large groups. All bacterial species, with a small exception, all bacterial species can be placed into one of two groups according to gram staining. One of those groups is called gram positive and one is called gram negative. And those classifications are based on the anatomy of the cell wall. Gram positive bacteria have a slightly different cell wall than gram negative bacteria do. And again, this is just a trait that bacteria have. Some bacteria are gram negative and some bacteria are gram positive. Just like some people are tall and some people are short, or some people have brown eyes and some people have blue eyes. Some bacteria have this gram-positive trait, and some bacteria have this gram-negative trait. The process for gram staining is not particularly difficult, and we're going to talk about it today, but it's critically important in getting us started down a pathway of identifying a particular species of bacteria. So that's what we'll be talking about today. Just a brief reminder of things on our schedule. I'm going to share my screen right now and pull up our course modules. Remember the to-do list for week 3, we talked about how you have 2 lectures to go through this week, prokaryotes and eukaryotes. Each one has a quiz associated with it. The last time we were together, I also recommended that you go on and do next week's lecture, if you have time, which is acellular microbes, viruses, and prions. If you have time to get that done, I recommend it because once you watch the acellular microbes lecture, you'll have all the lectures done for the next exam. And the next exam comes up next week. For laboratory, we did simple staining on Monday. We're doing gram staining today. You've got lab questions associated with that. And you have your first lab practical exam coming up next week as well. So remember, next week is the really busy week. You've got your next lecture exam. You've got your first lab practical exam. So you'll want to be sure that you draw out a schedule for yourself that is going to work for you so you can get everything done. All right. So does anyone have any questions about the simple staining material that we went through last time? Does anybody have any questions about making a smear on a bacterial culture, or heat fixing, or how we would stain that slide? The staining process is actually the easiest thing, right? You just put the stain on top of the smear and let it sit. And because that stain has a positive chromophore in it, it's pulled towards, it's attracted to the membrane around each cell, because that has a negative charge associated with it. And we end up with a nicely stained cell, hopefully. Remember, generally speaking, if you don't have other specific instructions, we leave a stain on a smear for about 60 seconds. That's a good rule of thumb. About 60 seconds should be plenty of time for that stain to adhere to the cell. Then we simply rinse it off, blot it dry a little bit, and then we can examine it under the microscope. All right, I'm going to go ahead and pull our slides up for today. That's what you should be seeing on your screen right now. You should see the title slide that says Lab Gram Staining. So our objectives today are shown on this slide. First, we're going to talk about the purpose, why we would bother gram staining a particular bacterial sample. We'll talk about the role of each one of the stains in this procedure. We'll talk about the actual procedure step by step. I'll demonstrate it for you. We'll talk about, we'll take a look at several different smears that have been gram stained, and we'll practice interpreting those. And we'll finish up today talking about some of the limitations of gram staining. In other words, the things that we have to keep in the back of our mind when we're gram staining that might impact our interpretation of the results. So what is the purpose of this procedure called gram staining? As I said a moment ago, bacteria can be divided into 2 large groups based on simply the anatomy of their cell wall. And that includes just about all bacteria, with one important exception, which I'll mention. We have gram-positive bacteria and we have gram-negative bacteria. Remember, this word gram is someone's last name. That's why it has a capital G associated with it. Gram-positive bacteria have a relatively thick layer of peptidoglycan around their cell membrane. Their cell wall is a relatively thick layer of this carbohydrate material called peptidoglycan. In comparison, a gram-negative bacterium has a thin layer of peptidoglycan. And then outside of that, they have a second membrane. So gram-positive cells have a thick layer of peptidoglycan, relatively speaking. Gram-negative cells have a much thinner layer, but they also have an outer membrane. And that outer membrane is quite different from their cell membrane. It just has a different anatomy than their cell membrane. So two very different general categories of bacteria. Now, when we complete the gram staining procedure, the result that we generate is called the gram status of the organism. And what the gram status is, or how it's reported, is whether the cell is gram positive or gram negative, and importantly, what shape the cells have. So for example, if I ran a gram stain procedure on a sample in the laboratory, I would report back to the physician or whoever else reported it, something like, this is a gram positive Caucus. Or I might say, this is a gram negative rod. So I'm going to report back whether it's gram positive or gram negative and what shape the cell has. Gram staining is the very first thing we do when we get a sample of unknown bacteria into the lab. The very first test we run is the gram stain. Once we place that unknown bacterium into a gram status group, then we can continue on with diagnostic testing in order to determine its identity. On this slide, you're looking at two drawings of the cell wall that we would find in a gram positive and a gram negative bacterium. Now we're only looking at a little piece of the cell wall. So you have to imagine that we just cut a little chunk out of that bacterial cell. And that's what this is over here on the right. Down here in brown, this represents the cell membrane. So here, this would be where the inside of the cell is, where the cytoplasm is, and the ribosomes and the DNA and such. Here's the cell membrane. Remember, it's a bilayer of phospholipids and proteins. Then there's a little bit of space, and then we find the cell wall. Remember, the cell wall in bacteria is made out of peptidoglycan. So that's what the purple lines represent, peptidoglycan. And in this drawing, the artist has represented a nice thick layer of peptidoglycan for us, gram positive. So out here, this would be the outside environment. This would be the inside of the cell in this drawing. Now take a look at the drawing on the left. Here's the cell membrane again in brown. Here's the peptidoglycan layer, the cell wall. Notice how thin it is. Much thinner than what you see on the right. Nice thin layer of peptidoglycan. But that's not all. We also have this. We have an outer membrane. We have a second membrane that's facing the outside world. This membrane is a little different than this membrane. The outer membrane, for example, has these strange looking green structures attached to it. This is a molecule called LPS, has the initials L, LPS. And LPS is important to the bacterium in terms of how it survives in its environment. We'll talk about LPS a little more later on. You don't have LPS in the cell membrane, but you do have it in the outer membrane because this is the membrane that faces the world. Gram-positive bacteria don't have an outer membrane, just gram-negative. Now, the gram staining procedure is a differential staining procedure, and it's also direct. So remember, we said a differential procedure uses multiple stains, and those multiple stains will stain different things, different colors. Remember that a direct procedure, that word means that we're actually staining the cells, not the background. Gram staining as a differential staining procedure has two really important parts to it. One is a decolorization step, where we're actually going to remove color, and what's called counter staining, where we remove one color and add another. That's what counter staining means. We're going to add another color to a place where we have removed some color. Now, the chemicals that we use in gram staining are listed right here. There are 4 of them. 2 of them are dyes, and 2 of them are additional chemicals. The first one is a dye. It's called crystal violet. Crystal violet is our primary stain. What crystal violet will do is adhere to cells and turn them a purple color. Some people describe it as more of a blue purple. You can make up your mind on that when you see that color. We're going to apply the crystal violet and stain the cells. Then we're going to rinse that off, and we're going to apply something called grams iodine. Now, this is not pure iodine. This is a modified iodine, hence grams iodine. And it's used in this procedure as something called a mordant. A mordant. If you've ever done any dyeing at home, you know that whenever we apply a dye to something like fabric or yarn or something, we have to do something to get the brightest colors possible. We have to add a chemical to make the colors be as bright as possible. And that chemical is called a mordant. What a mordant does is help that dye or that stain penetrate the material and stick. So it helps, in the case of gram staining, it helps the crystal violet penetrate the cell wall and adhere to it. If you didn't use grams iodine, you would probably still get some purple color, but it wouldn't be nearly as vivid. It wouldn't be nearly as bright and vivid. That's why we use that mordant, that grams iodine. So we put the grams iodine onto the smear, leave it for a while, and then we rinse it off. The third step in the procedure uses a chemical called a decolorizer. Now, the decolorizer is simply alcohol. And what the decolorizer does, like the name suggests, is it pulls some of the crystal violet out of the cell wall. Now, you would probably ask yourself here, why in the world would I go through this process and then remove the color? Why would I apply the dye, make sure that the dye penetrates and adheres, and then remove the dye? Well, here's the answer. We're not going to remove all the crystal violet. We're going to use the decolorizer for a specified amount of time, just enough time to pull out the crystal violet stain in just the thinnest layer of the peptidoglycan. Remember, the difference between a gram-positive and a gram-negative cell is how thick the peptidoglycan is. What our decolorizer will do is pull the crystal violet out of that thin layer of peptidoglycan in the gram-negative cell. It'll pull a little bit out of the gram-positive cell, but it will leave the rest in the gram-positive cell. So after this decolorizing step, the gram-negative cell is essentially unstained again, whereas the gram-positive cell remains mostly purple. So the key to decolorizing, as we'll talk about in a minute, is making sure you do it for the right amount of time and no more. The last step involves our second stain, and that's the stain called Safranin. If you remember from last time, Safranin leaves a beautiful sort of a fuchsia pink color. Safranin is our counter stain. It produces this vivid pink-red color in any cell that is unstained. So it's going to turn our gram-negative cells pink-red, while our gram-positive cells remain blue-purple. Take a look at this slide. This is a nice little step-by-step sort of visual diagram for us. Over here, the artist has drawn a gram-positive cell, and over here, a gram-negative cell. Now, he or she has made the gram-positive cell a caucus and the gram-negative cell a rod, but there's no significance to that. It could be any cell shape. So the first thing, remember, that we have to do is we have to make our smear and then fix it. We have to use heat or chemicals to perform the fixation step. Now we're ready to do our procedure. The first step, remember, is we apply crystal violet, and both the gram-positive and the gram-negative cells will turn purple from that stain. The dye, the crystal violet, is adhering to the peptidoglycan in the cell wall. The second step, remember, is the grams iodine treatment. That's our mordant. The mordant is going to help the stain penetrate and adhere to the peptidoglycan. Now, the artist has changed the purple a little bit here just to remind us that, we've done something. We pushed that purple color in deeply into the peptidoglycan, and we've gotten it to adhere. The third step is decolorization. And this is where the magic happens, because with that alcohol, that decolorizer, we're going to leave it on long enough to remove the crystal violet from the gram-negative cell, but not long enough to remove all the crystal violet from the gram-positive cell. Some of the crystal violet will leave the gram-positive cell, but because the cell wall is so thick in gram-positive cells, a lot of the crystal violet will remain. So the gram-positive cell, after decolorization, is still purple. The gram-negative cell is unstained. The last step is to apply the counter stain or the safranin. The unstained gram-negative cell will pick up the safranin and turn pink. The gram-positive cell will not turn pink because it's already stained purple. So when we look under the microscope after gram staining, if we see purple, we know we have a gram-positive cell. If we see pink, we know we have a gram-negative cell. If you think about it, it's an elegant little staining procedure. It's not that hard. It takes about 5 minutes, and it gives us a clear view without doing any additional testing of whether we have a bacterium with a thick cell wall, gram positive, or whether we have a bacterium with a thin cell wall, gram negative. Now, on this slide, all I've done is written down the exact procedure for gram staining, so you can have this when you study. I want us to be familiar with the chemicals that we are using for gram staining and what they do. I also want you to be familiar with the steps of the procedure, including the timing of the procedures. Now, in gram staining, most of these times that we use, for example, 30 seconds here or 60 seconds here, most of those times are approximate. In other words, if I apply crystal violet and I let it sit for, say, 35 seconds instead of 30, I haven't ruined anything. If I let it sit for 27 seconds, I haven't ruined anything. Approximately 30 seconds is fine. The only timed procedure in gram staining that's critical is the decolorizer step. So let's talk through this now. First thing we have to do is prepare and heat fix a smear. So we're going to spread out our cells, we're going to let them dry, and then we're going to use heat to help them adhere to the microscope slide. We're going to apply crystal violet over the smear and let it sit for 30 seconds. That's long enough for the crystal violet to stain the cell wall. Next, we're going to gently rinse off the crystal violet, and we're going to use distilled water. That's what lowercase d H2O, that's what that symbolizes, distilled water. Now, distilled water has had things removed from it. It's a way to sort of purify water. Can anybody think of a reason why I would not want to rinse off my stain with tap water? If you have an idea, put it in the chat. Why would I not want to use tap water here? All I'm doing is rinsing off the stain. So why use more expensive distilled water? Why not just put tap water on it? I mean, what's the big deal? I'm just rinsing. Why do you think I need distilled water? I've got some great answers here. Good job. Brooke and Sarah and Lisa and Candy and Maddie and Austin. Good, good. Tap water, while it's perfectly safe for us to drink in most instances, it's not a purified form of water. Number one, it's got chemicals in it. We know it's got chlorine in it, right? That's the disinfectant that's used in tap water. It's also got a lot of dissolved minerals in it. And it has bacteria in it. Now, it doesn't have a lot of bacteria in it, hopefully, but it does have bacteria in it. And the last thing we would want to do is contaminate our slide with those other things, especially other bacteria. Okay. You want to use a nice pure water because you don't want to add anything onto your slide. So hence the distilled water rinse. Good job. Now, while the slide is still wet from the rinse, you're going to put the grams iodine on top of the smear and let that sit for 30 seconds. Remember, this iodine is modified. It's had a couple things added into it to make it a mordant. After the 30 seconds, we're going to rinse it with distilled water. And now comes the decolorizing step. This is the one step that you have to be careful with your timing. We apply the decolorizer drop by drop you don't put a puddle of decolorizer on top of the smear. You actually apply it drop by drop over the smear for 10 seconds only. And we allow the decolorizer to run off the slide as we're doing it. Remember, this is removing some of the crystal violet stain. Then we gently rinse again with distilled water. Now we're ready to counter stain. We apply safranin in a puddle over the smear. And this one we're going to let sit for 60 seconds. So we counter stain longer than we use our primary stain. Then we gently rinse that off with water, distilled water, blot dry, and we're ready to examine our slide. So it's very straightforward. And like I said, gram staining is done in every microbiology lab every day. So whether it's a research laboratory or a teaching laboratory or a commercial laboratory or a diagnostic laboratory, gram staining is one of the fundamental procedures that's being done every single day. So there are people whose job it is in the micro lab to make all kinds of smears in the morning, many, many smears, and then gram stain them as a group, and then examine them and determine their gram status. All right. The next thing we're going to do is take a look at this procedure as it's done so you can see it visually. I like to see things visually. I'm sure some of you do too. Remember, the first thing we have to do is make our smear. And that's going to involve using our inoculating loop. You can see the wire from the inoculating loop in the picture here. Here's the loop at the very end. And I'm getting ready to sterilize my loop, as I do many, many, many times each day in the laboratory. Here's my incinerator device. I've got it turned on. It's nice and hot. Remember, I'm going to put the wire part of the loop down into the tunnel of the incinerator. This ceramic tunnel gets very, very hot. It gets red hot. And I'm going to allow the wire to glow red hot before I take it out and let it cool off. I'm going to use a pure culture of bacteria here. You could also, perhaps, depending on the situation, you could be using a sample that was collected from a patient, some kind of body fluid, in other words, that was collected from a patient. This is a pure culture of bacterium. This is broth, remember, liquid culture media, and there's bacteria growing in it. Notice I have a gloved hand. We always handle our culture containers with gloves on. Notice that I have tilted the test tube. When I open it, that's going to help keep bacteria from falling into my culture and contaminating it. Notice that I'm drawing the sample out from approximately the middle of the broth tube. Notice too that I am keeping the cap to the test tube in my hand. I don't want to put the cap down on the desk because I'll contaminate the cap. And when I go to put it back on, I'll end up contaminating the test tube. Next thing I'm going to do is take the fluid that's collected in the loop, and I'm going to spread it out onto the middle third of my microscope slide. Remember, we always label our slides, especially if we're planning to keep them. I just have a quick video clip here to show this process. I'm just making circular motions with this liquid. You end up making a smear probably the size of a quarter most of the time. You're just spreading that little bit of liquid out into as thin a smear as you can in the center of that slide. Now, remember, you've got to get your smear to dry before you can heat fix it. One of the tricks is just doing this. Just very gently with the slide at an angle, just waving it into the air. That sometimes can help a slide dry a little more quickly. You can certainly also just lay the slide down on a piece of paper towel, let it dry there. And remember, some laboratories have those drying trays that you can put a slide on. The trays that will gently, gently warm the slide and help them dry a little more quickly. Once the smear is absolutely dry, now I can heat fix it. I'm using the incinerator to heat fix it. This aluminum housing that's around the inner ceramic heating chamber, this housing gets very hot. So I'm using something to hold the slide. In this case, I'm using a clothespin. And that will allow me to lay the slide down on that hot housing without bringing my fingers near it. I'm going to set the slide right down on top of that hot aluminum metal, and I'm going to count between 20 and 30 seconds. You can see that I've made 3 different slides here. Again, like I said, this is very common when we're working in the lab. We rarely have one sample to gram stain. We usually have many samples that we have to gram stain. And there's no reason why you can't run those samples side by side and save yourself some time. So you can see all of these are labeled. You can also see here that I've drawn a circle with a Sharpie. Remember, that's also an option. It helps you find your smear under the microscope when you're just learning. So I would have smeared out my cells right inside that circle. And when I go to the microscope, I can use that black line as a guide to help me find my cells. Now, as I work through the gram staining procedure, I'm going to use this tool. This is a very low tech, very inexpensive tool that you will also find in microbiology labs. This is called a staining tray. If you remember when we were doing the simple staining last time, I showed you a picture of a staining bowl. You can certainly use a bowl to collect excess stain or stain waste, as we call it. You can certainly use a bowl, but when you want to do multiple slides at a time, it's nice to have one of these little inexpensive trays, because I can stain multiple slides at the same time using a tray. This is just a Tupperware or a plastic food container, and I've used 2 zip ties to create these sort of rails that I'm going to lay the slides down on. Here's one slide right here. I think you can just see it. Here's another slide right here. And here's the 3rd slide. So I can lay these down. I can stain all 3 of them at the same time. And I can also collect my waste material very effectively using a little tray like this. In this slide, you're looking at the set of chemicals that we use for gram staining. These were purchased commercially as a kit. Most laboratories do this. They buy gram stains as a kit. So this kit happens to come from Becton Dickinson, which is a commercial lab supplier. You can see you've got like a liter, I think, of stain in each bottle. If you're working in a laboratory that does lots and lots of gram staining, it makes sense to buy these chemicals in bulk. It'll save you a little bit of money. So step 1 is apply the crystal violet and let it sit for 30 seconds. So again, here's one slide here, and you can see I've covered the middle of the slide with crystal violet. I've made a nice big puddle of crystal violet. See that? Here's another one. Nice big puddle of crystal violet here. We draw up the crystal violet, sometimes with a transfer pipette. In some labs, they will put the crystal violet into a squeeze bottle so you can apply it directly. The important thing is that you cover the smear entirely. You don't want to be stingy with stain because you won't get good results. Next, I'm going to gently rinse the slide off. Notice I'm rinse the stain off, excuse me. Notice that I've picked up the slide with my clothespin again, this time just to keep the dye away from my hands. And I'm using a transfer pipette just to gently wash off the excess crystal violet. All that crystal violet and distilled water is just dripping down into my stain tray, so I'm collecting it. Notice too that I'm not squirting this slide. I'm not using any kind of force when I rinse off the crystal violet. It is super important that you're gentle with your smears. Don't be violent with your smears. That's because even though you've heat fixed those cells, you've gotten them to adhere to the slide. That doesn't mean that you can't remove those cells if you're too rough. If you do anything to sort of blast water down onto your smear, you can push some of the cells off the slide. Just like when you're done staining and you're blotting the slide, if you rub it, you can rub off cells. So we always blot. And when we're rinsing, we're always very gentle because you just made these smears. You don't want to ruin them. You want to keep them so you can examine them. So, Amy has a message. I hope you don't mind me sharing your question here, Amy, because it's a good one. And that is, can you stain multiple patient samples at the same time? Or do you have to stain one patient per go? And the answer is you can absolutely line up several different patients. This is a good example of why we have to be good labelers. If I'm going to take, let's say, 10 samples from 10 different patients, and I'm going to gram stain, it's going to save me time if I gram stain them side by side. So I'm doing them all together. I need to make double sure that I don't mix up anybody's sample with anybody else's sample. So when I make my smears, when I make my slides, I just want to be absolutely sure that I've identified the patient somehow, with a patient identification number or a sample number or whatever. You want to make sure that you know which slide belongs to which patient. But yes, you can absolutely do multiple diagnostic samples together. And that's because once you make a smear, once you've put cells onto a slide for gram staining, there's no way that you're going to move cells between slides. If you're doing it correctly, there's no way that you're going to contaminate one with another. All right. It's just that you're doing them at the same time. All right. I want you to notice that as I hold this rinsed slide up, I want you to notice that I can't see anything in this circle, can I? I don't see anything in here. I don't see any smear in here. I don't see the color purple in here. I just finished using my crystal violet, and yet I don't see anything in my circle. That's normal. That's normal. Remember, I put very, very thin layer of cells here. And that was done on purpose. I don't want a thick, thick layer of cells because I won't be able to interpret it very well. If you've made a nice thin smear, you're not going to see anything with your eyes because the stain has stained the cells, not the slide. You won't see the color until you go under the microscope. All right, the next step is grams iodine. That's what you're looking at here. I'm drawing up some grams iodine. I'm going to lay that in a puddle over the smears. Remember, grams iodine is not a stain. It's a mordant. It's used to help the crystal violet penetrate and adhere to the peptidoglycan. Grams iodine, just like crystal violet, is going to stay on top of the smear for about 30 seconds. And that's what you're seeing here. So like iodine that you would use as an antiseptic, it has this sort of deep amber brown color. And I'm just going to lay a nice heavy puddle of it over my smears. Notice my slides are still wet from the rinse I gave them. That's fine. That's not going to interfere with anything. I'm going to lay my grams iodine down and I'm going to leave it for 30 seconds. And then I'm going to rinse. Hold up each slide one at a time. Let the excess grams iodine go down into your stain tray. And then take your distilled water and gently rinse it off. Next step is the decolorizer. That's what you're looking at here. This is just an alcohol, basically. You'll notice that they've listed on the ingredients here that it's 3 parts of isopropanol alcohol, 1 part of acetone. Everybody's decolorizer is a little bit different. It depends on the company, but the bulk of a decolorizer is going to be alcohol because it's the alcohol that will draw stain out. Remember, the decolorizer step is the one that you have to be a little precise about. What you do, instead of laying a puddle of decolorizer like you do with the other 2 ingredients we've looked at, instead, you're going to hold the slide up at an angle over the staining tray. And using a transfer pipette, you're going to let go 1 drop at a time. And you're going to let that drop run down over the smear. You're going to let each droplet just run down over the smear and down into the staining tray. Remember what it's doing. It's pulling stain out of cells. So we want it to then go right down into our waste tray. I've got a little short clip here just to show you what this looks like. See how I'm dripping it down? I'm moving my pipetter back and forth so I make sure that the whole smear gets touched with the decolorizer solution. But I'm only going to drop droplets down for 10 seconds. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, done. 10 seconds, no more, no less. Now, it's really important that you rinse after you decolorize, right? You don't want the alcohol to sit on the slide and continue to decolorize. So you put drips down for 10 seconds and then rinse. You've done what you need to do. You have to trust. And then you finish. Now we're to our last step in the procedure, which is the counter stain. That's the safranin. The slides go back in the stain tray. We're going to put a nice big puddle of safranin over the smear. And remember, safranin sits for 60 seconds. So crystal violet for 30 seconds, rinse. Grams iodine for 30 seconds, rinse. Decolorize drop by drop for 10 seconds, rinse. And then safranin for 60 seconds. We do our final rinse, and then we gently blot. We blot those slides dry. They don't have to be bone dry. Just want to get most of the water off because it'll be easier to look at them under the microscope. And here are my slides. I'm ready to go to the microscope. I want you to notice a couple things. Notice that some of my label washed off, some of my circles washed off. If you use a regular Sharpie pen as your label to make your label, Sharpie pen will come off when it gets exposed to most decolorizers. So if you're not careful and you get a lot of decolorizer up here, you can lose your label. So just be careful about that. You can also use a different kind of marker. There's something called a grease pen. You might have used a grease pen in chemistry. Those are really waxy and the decolorizer doesn't tend to affect them as much. So you may lose a little bit of your writing, your Sharpie when you decolorize. The other thing I want you to notice is there's some bits and bobs of stain on these. See that? There's some purple down here and there's smudges of stain here and there. This is perfectly normal. Don't let that bother you. Don't think that you've done something wrong if you can see that. When you go through the gram staining procedure, it's not uncommon at all for your slide to look a little rough when you finish. It's still going to work for you. So at this point, I'm ready to go to the microscope. I'm ready to take my gloves off. I don't have any concerns that I might contaminate myself with bacteria because remember, these cells are dead now. They're dead. They've been heat fixed. They've been stained. They're no danger to me anymore. So it's okay for me to take my gloves off and touch these slides and put them under the microscope for examination. All right, any questions? Any questions about that procedure? It's pretty straightforward, right? You've got a nice little kit here. You've got everything you need in your kit. You just have to make your smear and you have to heat fix your smear, but then you're ready to go. Crystal violet for 30 seconds, rinse. Grams iodine for 30 seconds, rinse. Decolorize each slide one at a time for 10 seconds, drop by drop, rinse. Then you put the safranin on for 60 seconds, rinse. Then you blot it dry and you're ready to go. Brooke's asking a good question. Can you just use acetone? As you probably know, if you've ever used nail polish remover, if you've ever used a nail polish remover, which is pure acetone, acetone is also a decolorizer, right? But alcohol is a superior decolorizer. And it's because of the material that you're working with. If you were trying to pull color out of a synthetic material or a material that was that didn't come from a living cell, acetone alone would probably work. But because of the nature of peptidoglycan, because it's a carbohydrate material, the alcohol is the superior decolorizer. So again, it's going to vary from company to company, but all decolorizers are going to be primarily alcohol in the area of 70 to 80% alcohol, and then the rest maybe a little acetone. Some places put a little preservative in their decolorizer. It depends on the company, but most of it is just alcohol. Good question. All right, this is a good time for us to take a break. It's about 5 minutes to 10. Go get yourself a cup of coffee, stretch. I'll see you back in just 5 minutes. Okay, everybody, go ahead and type your name into chat for me again. Thank you so much. Thank you. I appreciate it. All right. So, does anybody have any questions about the procedure so far? Any questions about how you would perform the gram staining procedure? Again, it's pretty quick, not too difficult. Comes as a kit. All the materials come together as a kit, which is nice. Very, very common procedure. All right, what we'll do now is we will switch gears and we'll take a look at some gram stained smears, and we will practice determining the gram status of the organisms that we see. All right, take a look at this slide. What you're looking at on here is a mixed sample. One of the organisms on the smear is purple, and one of the organisms is pink. So on this one slide, on this one smear, we have an example of what gram positive looks like and what gram negative looks like. This is the purple color, the gram positive purple color, and this is the gram negative pink color. So those are the two colors that we're looking for when we evaluate a smear that has been gram stained. Now we are under the 100x lens here. So remember, that's 1000x total magnification. You take the power of the lens and you multiply it by 10, which is the power of the eyepieces. So 1000x, this is our best view, our most high magnification for cells. Now, what do you think about the shape? What do you think about the shape of the purple cells versus the shape of the pink cells? And no, it's not you. This particular slide, this image is a little fuzzy. So if it looks fuzzy to you, it's not your eyes, it's the slide. We need a shape for the purple cells, and we need a shape for the pink cells. Type it into chat. Good. Lisa's got it. Sarah, Crystal, Maddie. Good. Austin. Amy. Good. What we have on that particular slide are purple cocci. Remember, a caucus is one of those cells, and cocci are many of them. So purple cocci and pink rods. So if we were reporting this back, if we were writing up a report on this, these would be gram positive cocci and gram negative rods. Now, if you had any trouble with the rods, remember, you don't want to look at this when you're trying to determine cell shape. You want to look around and see if you can find a single cell. Here's one right here, and there's a second cell right next door. These are rods. These are bacilli, or rod-shaped cells. So there are two different organisms on this same smear. And as you can see, even though they went through the same staining procedure, they came out two very different colors. And that's because the caucus-shaped cells This is a species of bacteria that has a very thick cell wall, a thick layer of peptidoglycan. And the pink rod-shaped cells, they have a thin layer of peptidoglycan, and they took up that counter stain. What do you think about grouping? Do you see any grouping, do you think? Do you see any grouping for the cocci or the rods? Anybody see any particular groupings for the cocci or the rods? Are they forming chains or clusters? What do you think? I'll tell you, if there is grouping here, it's subtle. It's subtle, but there might be some grape clusters in the Caucus species. This might be some real clustering in the Caucus. Clustering is actually quite common, remember, for Staphylococcus. This could be a Staphylococcus organism. I don't see any obvious grouping in the rods. Now, I do see places where one or two rods have formed a chain. But if you have true chaining, you should see it all over the place. And generally more than 2 cells, 3, 4, 5 cells joining into a chain. So I don't think I see chaining in the rods. But certainly there's a couple of places where 2 cells have chained up, right? I will tell you this too. When it comes to cocci, it can be sort of a trap that we fall into sometimes when we start to think that cocci are gram-positive. Listen, a lot of the pathogens that we deal with, in humans anyway, a lot of the cocci type of bacteria are gram-positive. It just happens to be that way. But don't fall into the trap of thinking that if it's a coccus, it's going to be gram-positive. Similarly, don't fall into the trap of thinking that all rods are gram-negative. A lot of them are, but not all of them. So you can see anything. You can see gram-positive cocci, you can see gram-negative cocci. You can see gram-positive rods, you can see gram-negative rods, okay? All right. Here's another smear. This is another one that is not a pure culture. There's 2 different organisms on this smear. What do you think we have? Do you think you have gram-positive or gram-negative? And what shape do you have? Or shapes, because there's 2 organisms. Good, Brooke. Purple is gram-positive. Pink is gram-negative. Both of these organisms in here are rod-shaped. Remember, look around until you can find single cells. Here's one single rod. Here's a single rod. Here's a single rod up here and right next door to it. These are purple or gram-positive rods. And I also have pink, gram-negative rods. Now remember, if I look right here, it looks like I have pink cocci, right? Looks like I have pink cocci here. If you can look around and you can see rods, you have a rod. The reason you'll also sometimes see the Caucus look is because you're looking at some cells end on. Sometimes you're looking at the cell from the side and it looks like a rod. Sometimes you're looking at it from the end and it looks like a Caucus. All right. If it's a Caucus, you won't see anything shaped like a rod. You see what I'm saying? If any of the cells are rod-shaped, it's a rod. Okay. If it's a real caucus, then they should all, sorry, I'm trying to do this. They should all be caucus shaped. They should all be circular. So again, I've got 1 gram positive rod and I've got 1 gram negative rod here. All right. On this slide, I've got a couple next to each other. This is a gram positive caucus. This happens to be underneath the 40x lens. So that's a 400 times total magnification. I just want you to look at this. You can see this, right? You can see purple and you can also see cocci. You can see circles. You don't see them as well as you would at the 100x, but you can see them, right? You can see bacteria at 40x. And if they're stained with the gram stain procedure, you can determine the gram status at 40x. Gram positive caucus, gram negative rod. Notice the difference though at 40x and 100x. You can just see things more clearly under the 100x lens. The cells are that much bigger. And so it's much easier to look at them and determine what the shape is under the 100x lens. But this is what I mean when I tell you, you can see bacteria under 40x. It's just that they're a lot smaller, a little bit harder to see the shape. This one happens to be a Staphylococcus. This one happens to be a Bacillus. Remember, you cannot identify bacteria from gram staining. I can't look at this and say, oh yeah, this is definitely Staphylococcus aureus, or this is definitely Bacillus subtilis. I can't do that. I don't have enough information. All I can tell you from a gram stain is the gram status, gram positive caucus, gram negative rod. Think about this slide. Think about what this looks like under 40x and compare it to this one. This is 100x. It's quite a difference, right? The cells are just, they're bigger. They're easier to see. This is 40x. This is 100x. What do you think? What's the gram status? What's the gram status here? Type it into chat. What's the gram status of that organism? Here it is again. What have I got here? There's one organism here. This is a pure culture that was grown in the lab. What are you going to write in your report? Good. Good. Now remember, and I'm going to be a stickler about this on lab practical exams, we report this as gram positive or gram negative cocci. You're going to write the word gram and then positive or negative, and then you're going to tell me the shape. That's the status. That's the gram status. Gram positive caucus or gram positive cocci. Okay, good. Here's your next one. Now, this one is also under the 100x lens. This came from a diagnostic lab. This was a patient sample. This sample was a swab from the back of a patient's throat. Remember, when we have a pure culture, in other words, when we get the cells from a culture, that was grown in the lab, the background is going to be pure white. It's going to be just nice and clear and white. But when you take body fluid and you make a smear from it, the background is going to pick up some color. So the material from the back of this person's throat had also picked up some stain color, mucus, and so on. But here's my organism, right? Here's my organism growing. First of all, tell me the gram status. What's the gram status? When am I going to tell that physician about the bacteria growing in the back of that person's throat? Lisa's got it. And Candy and Amy and Brooke and Austin. Oh, Austin's only getting half credit. Sarah's got it. Maddie's got it. Oh, Amy's getting half credit. Candy's getting half credit. This is a gram-positive Caucus. Don't forget to tell me what cell shape you see. This is a gram-positive Caucus. And this Caucus is forming some very nice chains. See the chaining? Look at these chains. This organism is living in little communities of cells. They join together end to end. See those? In fact, there are very few that are actually individual. I see a lot of couples. I see a lot of 2-cell chains, 2 cells, 2 cells, and then a lot of these long chains. Okay. Gram-positive Caucus. We would also, to give Brooke a little extra credit here, we would also tell the physician that there's chaining present. There's chaining present here, because part of what the physician is going to do is use the information that we provide to make an educated guess about what organism this is. Remember, we cannot tell the physician what this is. We don't know. We don't know what genus and species this is. We haven't done enough testing yet. But we can tell the physician that it's a gram-positive Caucus, and we can tell them that it's forming chains. And you're right, Lisa, this happens to be a Streptococcus. Streptococcus would have been, for the physician, would have been a reasonable guess. Number one, because where this sample came from. This patient had a sore throat. And one of the most common causes of sore throat, at least bacterial, is a Streptococcus called Streptococcus pyogenes. That's a cause of bacterial sore throat. You've probably heard of strep throat. That's what people mean when they call it a strep throat, Streptococcus pyogenes. Now, you don't know that it's strep pyogenes, though. You don't know that because you haven't finished your diagnostic work. But it would be perfectly reasonable for the physician to guess that this is probably strep pyogenes and to choose an antibiotic that will kill strep pyogenes. Perfectly reasonable to do that. And that's what happens most of the time in the first case of a bacterial infection. Physicians treat empirically. In other words, they treat based on what they think the bacterium is, not what they know the bacterium is. Now, if something weird were to happen, like for example, let's say the physician gave that patient an antibiotic and the sore throat didn't go away, or the sore throat got worse, or maybe the patient developed additional symptoms. At that point, the physician might want to take a second sample and submit it for culture and identification. because at that point, it's not behaving the way we would expect it to behave. So yes, physicians do order sometimes culture and identification. They want us to determine what the genus and the species is. But a lot of time, most of the time, a physician will treat a patient by using empirical diagnosis. In other words, guessing based on the evidence what that organism is. So don't forget, when you tell me what the gram status is, tell me gram positive or gram negative and the shape of the cell. Take a look at this side by side. We're under a 40x lens here. This is a pure culture of cells. This came from a patient sample. Do you see the difference? I would sure rather have to interpret this than interpret this. I mean, I guess this isn't hard to interpret, but there's a lot of stuff on here that is not the organism. Everywhere I see purple here, these purple circles, this tells me that this is a gram-positive Caucus. But you can see where there's also some just pink, this pink material in here. Over here, it's very different. Over here, all I see is purple, purple sitting on a white background. All right. It's hard to tell what the shape is over here. Much easier to tell over here because here we're under a higher magnification. Now, here's something I want you to remember for me. When you look at a slide that comes from a body fluid, and you see, you say to yourself, I definitely see cocci here, and they're definitely purple. So gram-positive Caucus. But I'm not sure what the pink is. Maybe the pink is also a Caucus, because I think I see some circles. I think I see some circles. But of course, I also see stuff that's not circular. I see stuff that's not very symmetrical at all. That's also pink. If you are looking at a bacterium on a gram stain, if you're looking at a bacterial organism that has picked up either purple or pink, because those are the only two colors they're going to pick up, you're going to see them all having about the same shape. And remember, living things, living cells are symmetrical. So anytime you see shapes on a smear that are not symmetrical and are terrifically variable, so, you know, I might have this piece that's sort of crooked, that's little, and then I might have another piece that looks like a big oval with some spikes coming out of it. Those aren't cells. Those aren't cells. That's debris. That's just debris on the slide. Remember, if it came out of a patient's body somehow, there's going to be material that comes along with the cells. If I, for example, collected some material from an abscess, or if you're not familiar with that word, think about a really large pimple somewhere on the skin. A collection of white blood cells and bacteria and debris. That's what pus is. Pus is a collection of things. And when you put it on a slide, when you make a smear from it and you gram stain it, there are going to be bacteria in there, but there's also going to be other stuff in there. So it's your job to figure out what are actually cells and what is just debris. Cells are going to be all the same size, all the same shape. Debris is not. Debris is going to have varying sizes and varying shapes. But debris will pick up stain. Remember, debris has maybe protein in it, maybe carbohydrate, who knows? And it will pick up stain. So it can be a little hard. It can be a little hard when you're looking at an actual real world sample from a patient. It can be hard. And it takes practice to figure out what is a cell and what is just debris. Be also very careful when you're taking an exam, be careful. Not all smears are gram stained smears. I have to tell you it's a gram stain, because I can also just maybe make a simple stain. I can turn cells purple using a simple stain. I can turn cells pink using a simple stain. You can only tell me the gram status if I tell you that that smear has been gram stained. Oh, just so you know, this happens to be a Staphylococcus organism. This is one called Staphylococcus epidermidis. It's a very common organism on the skin, and it can be a pathogen. And yes, you do see some grouping. You see some grape clusters here. Again, typical of a Staphylococcus organism. All right, here's my next smear. What do you think? Gram positive? Gram negative? What's the cell shape? Good job. Brooks got it. Casey's got it. Sarah, Candy, Elizabeth, good. Amy, good. Maddie, good. Yes, we have gram positive rods here. And yes, you do see some chaining here. Good job, Lisa. Yes, gram positive rods. We're under the 100x lens here, I believe. I'm not under 40 here, am I? Hold on, let me double check. No, I am under 40. This is a 40x. And look at how clearly you can see these rods, right? We're under the 40x lens. Do you remember I showed you these cocci are under the 40x lens? These rods are under the 40x lens. These rods are huge. This is that organism called Bacillus megaterium. This is a giant rod. Just a big, big rod, and it's gram positive, and it likes to chain. So cocci are not the only bacteria that like to form chains. This is a side by side. Here's one under the 40x lens. Here's one under the 100x lens. Same organism. What do you think? Gram positive, gram negative, shape. Gram positive, gram negative. You can tell under the 40x. But it sure is easier under the 100x, isn't it? It's just easier. What do you think? Yay, everybody's getting it. Hooray. Gram negative rods. Pink is gram negative, and those are rods. I tell you what, though, those are some stubby little rods. This is E. coli. Remember, try to find a single cell. Try to find one cell. There's a group of cells here, but you can see them individually. These are little short stubby rods. That's characteristic of E. coli. Look down here. I can see two rods end to end in a chain. Look at how short they are. That's very characteristic of E. coli. Compare that rod to this rod, which is much longer. Here's two rods end to end. E. coli is known for being sort of stubby and short, and also gram negative. So again, we can look under the 40X and we can get a gram status. But if you look here and you don't feel good about whether you see rods or cocci, you just go up to 100X and you'll get a much better view. I want you to also notice that there's no real, there's nothing going on here in terms of grouping behavior. You might think, oh, well, maybe these are clusters. Maybe these are clusters. Rods don't tend to cluster. Rods will change sometimes, but they don't cluster. Cocci cluster. But rods don't typically. And E. coli doesn't have any particular behaviors, grouping behaviors that we look for on the slide. So again, if I saw that smear, I would report that that is a gram-negative rod. I might even report that it's a short rod. I would not try to identify that organism because I don't know what it is. I don't know. I haven't done enough tests yet. I don't know that that's E. coli. I haven't done enough tests. The physician might make an educated guess that it's E. coli. The physician might. If the physician knows what symptoms the patient has, what sample this came from, and the physician has an idea, I think it might be E. coli. The physician will probably treat for E. coli. But we don't know that it's E. coli yet because we haven't done enough testing to identify it as E. coli. Yeah, so compare the size. We're under the 40X lens here, E. coli. We're under the 40X lens here, Bacillus megaterium. Look at the difference in the size of those rods. Megaterium got its name because the rods are so big. It's just a big cell. It's a big organism. All right. One of the things we need to know about gram staining is what its limits are. And this is really practical information more than anything. Gram staining is best accomplished on a fresh bacterial culture or a fresh patient sample. And what do I mean by fresh? In microbiology, I mean less than 24 hours old. If I grow a culture in the incubator for, let's say, 48 hours, those cells are getting old. And what will happen is if I try to gram stain that culture, that 48-hour old culture, what's going to happen is I'm going to get inconsistent staining. Now, what do I mean by that? If I take a pure culture out of the incubator at 48 hours and I make a smear and I gram stain it, what's going to happen is the cells are going to show me a little purple and a little pink. Inconsistent staining. Some cells might be completely purple. Some cells might be completely pink. Some cells might be half purple and half pink. I'm going to see inconsistent staining. If I know that I have a pure culture, if I know that there's one organism growing in that culture and I get inconsistent staining, I can't interpret anything on that. I can't determine the gram status on that. I don't know. Are the pink cells accurate? Are the purple cells accurate? I don't know. If you see inconsistent staining, you cannot determine the gram status. Nope, that culture's too old. Now remember, all is not lost at that point because we can make a subculture. We can take a few cells, we can take a small volume out of that culture tube or that culture plate, and we can put it into some fresh media and put it back in the incubator. And this time, take it out at 24 hours and do a gram stain. So if you end up with an old culture and it doesn't stain consistently, we don't give up, we just subculture and we repeat the gram stain, and hopefully we'll get a better result the second time. So gram staining is best done on fresh cells, less than 24 hours old. When a physician takes a sample from a patient and sends it to the laboratory for gram staining, it's really important that that sample get to the lab quickly. And that's why in say a doctor's office, the samples are picked up typically every single day, right? Someone from Quest Laboratory or someone from LabCorp or whatever is going to come and pick up those samples and get them to the lab. And that testing needs to be done relatively quickly. I can't let that sample sit around for a couple days and then try to gram stain it. It's not going to work. The cells are going to get old and they're going to start dying, and they're not going to pick up stain consistently. Now, if the organism that I'm trying to gram stain, if the organism growing in my culture happens to be a bacillus or a clostridium, if I've got an old culture of a bacillus organism or a clostridium organism, not only is it going to stain inconsistently, but I'm probably going to start to see sporulation occurring. Okay, so it's going to gram stain poorly, inconsistently, and I'm likely to also see some signs of sporulation. If you ever do a gram stain and the cells show signs that they're sporulating, your culture is too old. Your sample is too old, and you cannot trust those results. You need a fresh sample. You need a fresh culture. So the age of the cells really matters with gram staining. Here's something else to remember, very practical. Gram stains themselves have expiration dates. In fact, all stains have expiration dates. And when we buy stains, we have to be sure to circle that expiration date on the container to remind us when it is no longer fresh enough to use. Old stains have to be discarded because they just don't work. Fresh stains are the best. So those kits we buy, the gram staining kits we buy, they're going to have an expiration date. And in my experience, they usually last about a year. If I buy a gram staining kit from a commercial producer, I'm probably going to have an expiration date about a year out. And what that means is I can use those stains for a year, which is great. But once they hit their expiration date, you have to discard them. You can't trust them beyond that point. You cannot trust that those stains are still working as intended beyond that point, because the chemicals will change with time. And they're just not trustworthy. So you have to put those old stains into your hazardous waste, and you have to order some fresh ones. So those are the two big limitations: the age of the cells and the age of the stains, essentially. Fresh cells and fresh stains are best. Take a look at this smear. This is a smear that was made on a culture that was getting old. Take a look at this and think about the other ones we've seen today. Think about what we've seen in terms of smears made from pure cultures. Nice white backgrounds. You don't see a lot of debris. Nice even staining of cells. And then look at this one. Now, at first glance, and I'm under the 100 excellence here. At first glance, you might say, oh, gram-positive Caucus. Gram-positive Caucus. I see purple circles. And they're clustering. They're clustering. Gram-positive Caucus. You'd be forgiven for saying that as a student, as someone who's learning. But with a little experience, you would stop and you would say, hold on a minute, this doesn't look like a good smear to me. Number one, there's a lot of debris on this slide. Look at this material up here that's almost black. I've got a great big pink circle right here. I've got a great big black circle right here. I've got a small black circle. I've got more pink stuff. There's stuff on here that is not cellular. There's debris on here. And if this came from a pure sample, a pure culture, let me say, in other words, a container that I made in the laboratory that should have one organism growing in it, and I see all this debris, right away I'm going to think, uh-oh, I think this culture is getting old. I think I might have some dead cells and just other material floating around in here, waste material floating around in here. The other thing I'm starting to see is something like this. I can see some circular cells right here that are pink. I see some cells down here that are pink, not purple. If I look around, I start to see pink cells. There's some pink ones right there. Huh. Now, if this was from a patient, if this came from a patient sample, the debris would not be unusual. If this was a mucus sample or a throat swab or, you know, from an abscess or some other material that came directly from a patient, seeing debris would not be unusual. No. And if I saw purple and pink cells, part of you might say, well, maybe there's two different organisms here. Maybe I have a gram-positive Caucus and a gram-negative Caucus growing. Here's the thing. That would be highly unlikely. When a human being gets an infection somewhere, it's most common to have a single organism causing that infection. It's very uncommon for a human being to have an infection caused by two different organisms. So yes, that would be very odd to have both gram-positive and gram-negative cocci on a smear. It's much more likely here that this is an old sample. And again, I told you it's pure culture. These are old cells and they're staining inconsistently. Some of them are staining purple. Some of them are staining pink. And I've got a bunch of debris. If this is a pure culture, this is a culture that I cannot interpret this result. I would have to say, for gram status, I would have to say unable to determine. I suspect this is an old culture. See that? Inconsistent staining. By the way, this happens to be a Staphylococcus culture. So I would expect it to be gram-positive because Staphylococcus is a gram-positive organism. I can also see clustering here that's characteristic of Staphylococcus. But if I were looking at this smear, I would not be able to determine gram status because I see inconsistent staining and I see a lot of debris. Here's another good one. Take a look at this. Again, pure culture. These cells were grown in a culture container in the incubator. See how inconsistently they have picked up stain. They were gram stained. This smear was gram stained. And boy, oh boy, I don't know what I've got. Look at this rod right here. It's purple at the ends and pink in the middle. I've got some purple rods here. I've got other ones that are pink, that are completely pink. This is a sporulating cell. Now, how do I know it's sporulating? There's a thing in the middle that hasn't picked up any stain at all. It's white. There's another one right here. That's a spore. It's white. It hasn't picked up any stain. This spore, this culture is starting to sporulate. Here's a spore right here. That's white. Yikes. What's going on? Remember, anytime that you see a smear that was gram stained, and if it's bacillus or clostridium, it's able to sporulate. Anytime you see a smear and there are cells sporulating, a gram stained smear, and there are cells sporulating, don't try to interpret that. That culture is old. That sample is old. The only reason that bacillus or clostridium bacteria would sporulate is if they were under stress. There's not enough water, there's not enough nutrients, there's too much waste. They're under stress. They're starting to sporulate. That means I just can't trust the results of that gram stain. Some of the cells are pink, some are purple, some are both. I can't interpret that. So Brooke is asking a great question. If a sample is fixed, in other words, put into something like formalin, if a tissue sample is fixed, does the 24-hour period not apply? That's a great question. So the only difference between what we've been talking about, Brooke, and what you're talking about is what the sample is made out of. If you're collecting, say, a throat swab, or a sputum sample, or a urine sample, or a blood sample, if you're collecting a bronchial sample, fluid from the bronchus or the trachea, those materials can't be fixed. We can fix tissue. We can fix biopsy samples. And yes, the 24-hour would not apply if, for example, I got a biopsy from a patient's stomach and it was fixed. Now the cells have been put into, again, a suspended animation. They're dead because formalin will kill them, but now they're stuck in time. Here's the thing though, I would not be gram staining a fixed sample. Now, I know I told you that gram staining is like the first thing we do, but you need living cells for gram staining. Once you fix a cell, whether it's a eukaryotic cell or a prokaryotic cell, you have altered its chemical nature. So if I got a biopsy, what I would do is prepare a thin section of it, and I would look at it under the microscope, and I would perhaps see the bacteria. I would report that back. I would say something like, there are rod-shaped bacteria in here. And then the physician would interpret it however they want to interpret that. But I would not be gram staining that because the cells are no longer fresh. The cells have been fixed. They have been altered in their nature. That's a great question. And now Amy's asking, do the people who are collecting these samples report the time when the sample was taken? Amy, they are supposed to report the time. Let me tell you from my own experience in veterinary clinics, we don't always report the time, but yeah, you're supposed to. You're supposed to say, you know, there are forms that you fill out. Each microbiology laboratory will have its own form that you'll submit with your sample. And it will ask you, you know, the identifiers for the patient. How old are they? What species are they? What are their symptoms? Where did you collect the sample? When did you collect the sample? Yeah. So yeah, ideally we know when the sample was collected so we know how old the cells are. Yeah. Now that doesn't mean that they all do it, believe me. Yeah, especially, you know, in a busy emergency department, maybe not. The samples may go upstairs without much information at all. But yeah, ideally we mark that down. This happens to be, just for interest's sake, this happens to be a bacillus organism, hence the sporulation. It happens to be Bacillus subtilis. These are smaller rods. We're under the 100x lens here. These are smaller rods than that Bacillus megaterium organism we looked at. All right, let's do some clinical work here. This was a throat swab from a patient that was complaining about a sore throat. Made a smear, heat fixed it, gram stained it. Take a look. What's the gram status of this? What are you going to tell the physician? Go ahead and type that into chat for me. This patient came in complaining of a sore throat. Doctor took a swab, sent it to the lab. We made a smear. What do you think? What's the gram status of this organism? Good, Brooke. Good, Lisa. Amy. Good. Gram-positive Caucus. And I see some chaining present there too. Good, Elizabeth. Good. Good, Austin. Gram-positive Caucus. Very good. Now, what's all this pink material? I see some pink stuff right here. I see a sort of a long, I don't know, string of something that's turned pink. I see some pink stuff over here too. And some pink stuff down here. What is this? What's all this? Yuck. What's that right there that's pink? I see some pink over here too. In fact, the back of the slide looks pink. What's all that? Yeah. Amy's got it. Brooke's got it. Elizabeth's got it. That's debris. Remember, this patient has a sore throat. This patient's got an inflammatory process going on in their pharynx. They've probably got some mucus back there, right? With some strands of fibrin in it, maybe. You might even occasionally pick up, you know, a white blood cell or two. Right. It's debris. It's debris. The organism is what's purple on that smear. It's a Caucus. It's purple, so it's gram-positive. And it is forming chains. Turns out it's a Streptococcus. Very common cause of sore throats. Streptococcus pyogenes. Very treatable with antibiotics. That's the only good thing about having strep throat, is that within a day or two, you'll feel a lot better if you take your antibiotics. Who's got questions? Any questions at all? Gram staining is a very important procedure in the micro lab. It's the very first thing we can do to help identify an organism. We can break it down into one of two groups, gram-positive or gram-negative. And then we can move on from there and choose some diagnostic tests to help identify the genus and the species of the organism. Now, I mentioned right at the beginning that there is an exception to gram staining. There's an exception to being put into one of those two groups. There are some bacteria that can't be gram stained. They can't be gram stained. They will not pick up the gram stains. And we talked about this in lecture. This is the Mycobacterium. Mycobacteria are gram indeterminate. Austin wrote that down in our chat. Gram indeterminate. Mycobacteria are a genus of bacteria that secrete a molecule outside of their cell wall. It's a waxy material. And that waxy material prevents the gram stains from penetrating and staining the peptidoglycan. So you cannot gram stain mycobacteria. It just doesn't work. What you have to do instead, if you think you have a mycobacterium, is you have to do a procedure called an acid fast staining. It's a different procedure. It will help dissolve away that waxy material, and it will stain the cells. The most famous mycobacterium is Mycobacterium tuberculosis. That is the organism that causes tuberculosis in humans. If I got a sample in and I tried to gram stain it, and when I looked under the microscope, I didn't see any color at all. First of all, I'd probably repeat it just in case I had made a mistake. But if I saw colorless cells under the microscope, I would be very suspicious that I had a mycobacterium. And the way I would double check is I would do an acid fast staining procedure and see if I could color those. Mycobacteria are unusual in that they produce that waxy material. They look very different when they grow on an agar plate as well. And we'll have a chance to see them in a week or two when we look at some colonies growing on agar. Very interesting organism, causes a very bad disease. So yes, my report back to the physician would be gram indeterminate. And I would recommend acid fast staining. Good question, Austin. All right, any other questions? All right, folks, we're almost out of time. You're all set now to do your lab homework for the week. I recommend getting that in sooner rather than later, because I can get it graded and returned to you if you submit it. Remember, you have a lab practical exam coming up next week. So again, I would recommend getting your lab homework done sooner this week rather than later. We'll talk a little bit more about the nuts and bolts of the lab practical when we get together on Monday for lab. Okay, remember, you have 2 quizzes this week, prokaryotic cells and eukaryotic cells. If you can do the acellular microbes lecture, I highly recommend it. Get it done, get it out of the way, take that quiz. It'll free up some time next week for studying. All right, everybody. I'll see you again on Monday morning. If you have any questions before then, just message me through Canvas. Have a great rest of the week.